Fritz for organizing this and inviting me to this. So this is much more basic than what you have seen so far, so set your expectations accordingly, because as you will see, the road that we have to go to get to the level of some other applications is quite long in pollinator monitoring. So first, well, my talk is divided slightly in 4 parts, but let's get started. I also will not probably fill the whole hour because I want to leave plenty of time for questions. So just so you know, okay, so first of all, we can study diversity at many different levels. So from the smallest one, from the molecular, from molecular biology, genes, to the genetic diversity interactions and geography. So historically, genes have received a lot of attention because since the discovery of DNA, there have been like sort of a unified approach to study this information. And what I think is crucial is that they realized pretty quickly a format for their data to be in, so that they can all agree and work on the same kind of data, in which case is text, which is also easy to manipulate, but that's another thing. Species occurrence data has also seen a renaissance with the species distribution models. This has been spurred by remote sensing technology and all sorts of geographical processing systems, and as well as camera traps. That we've seen some examples already on how ecologists have been using camera traps to fit the distribution models and study species occurrences. And it's emerging the application of eDNA to study on a broad scale, but at a somewhat low taxonomic resolution, the species occurrences with less invasive methods. We cannot say quite the same that we found like a standardized set of data that everybody wants to use and would like to study for functional traits and interactions. I would say that with organisms interactions, we're even farther from this than these other technologies. So, of course, technology has been advancing steadily, and we have many instruments that can collect an array of data in the wild. And as we've seen that from lab setups that are relatively controlled and take images and some other kinds of data, but these kinds of sensors can also be deployed in the field, and in the field you don't have quite as controlled conditions, so your data tend to be more noisy, and the signal to noise ratio is also worse. So the methods that you apply in the lab not always transfer super well when you actually try to do it in nature. So yeah, my talk focuses mostly on images because this is a computer vision forum, and I feel like the processing of the images is one of the most severe bottlenecks. The acquisition part is also complicated. It has its own drawbacks, as we will see later, but the processing is also like it's strongly influenced by the methods that are available and the availability of expertise in the discipline. So yeah, I mean, I probably don't need to convince you, but artificial intelligence is probably a good idea to be applied when we have these big amounts of data. Right. So my first part, I call it phenomics, but it's really computer vision, pure computer vision. So we often collect, as biologists, we collect images, and oftentimes we want to measure something on them. This something is, in a lot of cases, is traits. And here I present some example of images that are collected in highly standardized settings, which are highly repeatable, and their signal to noise ratio is quite high. This can be done, of course, for plants, and here I have some classic Arabidopsis like morphotypes, as well as some more like complicated and maybe evolutionarily significant macrophotographs of modular flowers, so that you can study the evolution of the modularity of the flower, of the Asteraceae of this family across the evolutionary tree by measuring traits on them. But this can also be done, of course, on insects. For instance, there is quite a bit of work done on wing venation and how it does relate to phylogeny. So you have to remember there is always a trade-off between image standardization and how much time you need to measure it. The less time you spend on acquiring the images, the more time you will spend to acquire a great level of detail in your images. If you spend a lot of time acquiring images, then they're highly standardized, and then your post-processing is faster and more accurate. I will also point out that in my opinion, the vast majority of disciplines, still in sub-disciplines in biology, the measurements are still done by hand. Like people take a picture, then they have a scale and use ImageJ or another equivalent tool and just measure things on the image. So yeah, but why do we even want to measure traits on images? Because traits are important to mediate the relationship between the organism and the environment as well as other organisms. We can also, as Eric also pointed out, we can also measure selection directly on the traits without even knowing the genetic basis underlying them. This approach is called the selection gradients, and has been a staple in evolution, in classical evolutionary biology for a long time. So, yeah. Okay, I probably don't need to tell you this, but repetita juvant, we say. So, computers store the images as digital images are stored as a matrix, and this is basically a big array of numbers. And computer vision, also called signal processing in some domains, uses simple mathematical operations on these matrices to highlight certain features. Like this, in this case, for instance, I'm showing like a simple thresholding algorithm, and you can also apply some other filter that can basically smooth the image to then apply some other thresholding algorithms afterwards. So these are all very simple techniques that have been around for a very long time, and they still can give meaningful results for biological images. All right, so this is the first part of the project that I'm gonna talk about. It's called Biodetect, and we want it to be, and it's a collaborative project between the Swiss Data Science Center. This is also part of the ATH domain, VSL and EVAG, which are two research institutes in Switzerland. So we wanted to do two things. One is using highly standardized images, like we have seen a lot of, to measure some traits, and the other one is training a detector for visitors to focal objects, but we'll get to that later. So let's get started on the Macrozobenthos phenotyping tool. So Macrozobenthos are invertebrates larger than 1 millimeter, living on the bottom of water bodies. Their species presence, abundance, traits have been used for assessing the environmental quality and biodiversity. There is quite a big corpus of literature on how these two interact. And importantly, a lot of stakeholders that monitor water quality, environmental quality, will use this kind of these organisms as indicators. So there is like a direct application when you analyze these organisms. So yeah. Still currently, to this day, Macrozobenthos is collected manually and manually sorted in taxonomic groups and measured for this very purpose. So the data that we collected derives from, not only, but mainly from an experiment that manipulated the flux in a freshwater system to basically assess whether the increased or decreased flow, for instance, for before and after an hydroelectric dam, will impact the composition and size distribution of the population of these animals. So the camera setup that we used has been fairly highly standardized, and there is quite a big amount of work that went into standardizing the way that we take images. But the important part is that the camera is always at the same distance from the tray where you take the images, and that we try to not have overlap of individuals to make our life easier in post-processing. So, yeah, so these are some of the images that we are taking. As you can see, it's on a white background, uniform illumination, there are very little shadows, and we have a scale on always in the same position in the bottom right. This is always to make our life a little bit easier. But as you can see, there is a variety of body sizes, of shapes, and different also colors of the organisms, even though these are in alcohol, so they all kind of look shades of brown. But the important part is that the signal to noise ratio is quite high. So, right, so this is the first package that we prepared. We made it as a modular Python package for high throughput analysis of this kind of images in the lab. So we tried to make it as simple as possible for somebody that is not very used to coding to have a minimum amount of command line interaction and still be able to run from the image directly to the result, basically. But each of these separate modules in this pipeline can also be run independently, plugging in your data and grabbing the result of it and doing something different with it. everything is open source, so you can see the code, the source code and modify the functions and everything. We also provide a pre-trained classification model on macrosegmental samples for Switzerland. It goes only to a relatively coarse resolution because they're difficult and we don't have a ton of data to train them on. But the most important part about this is that the user can provide their own labeled data, and with basically one line, the pipeline will retrain the model based on the user data. And this can be done on CPU in a reasonable amount of time. We also provide a way to automatically extract the length and the head width of the organisms in two separate ways. So yeah, so this is the pipeline. Here I show just how some parts are unsupervised and don't need any training or any training data, so they should be, when you fiddle around with the parameters, you should be basically able to obtain good results without providing any training data. But other parts, of course, like the classification are based on deep learning models, so that will need to be trained. So on these, I will make the point of showing you. So this is the documentation of the package, and I make the point that I probably spent 75% of my time writing the documentation and maybe 25% coding the rest. And this is really important because if it's not well documented, people will not use it. So when you download this, you can download it as, let's go to installation. You can download a Docker container. So you install your Docker engine, which is fairly easy. Everybody should be able to do it. You download the Docker container, and when you launch it, you drop exactly into the same environment that I programmed it without basically any possibility of messing up the environment or unlinking the data and things, stuff like that. So yeah, you can also install manually and import the functions from the different functions from inside the module. I go in painstaking detail in all of these, like I explain everything that happens in the package step by step, basically as ChatGPT would have explained it to you if you gave it the code. I've done this before, but too bad. And yeah, so there is really quite a lot of detail, and basically the way you interact with it, you only need to change the location of your files and a few other parameters that are all stored in a giant configuration file, in which, don't mind the bold, this is an old version, but you can basically tweak the parameters of all of these that are explained in detail, and you should be able to get the results that we show in the examples pretty quickly with a data set that this package has never seen, and yeah. So this was like the basic idea for the architecture of this modular package that we hope to be reusing also for other software packages that we will be releasing in the future. But let's look at some results from this. So here, from the big images that I showed you before, the package has basically extracted the clips, has saved an RGB and a binary mask, the one in black and white, and has also measured you can see the green line that is basically the length of the organisms, and the width. This is actually a little outdated because now it measures actually on the head and on the middle of the organism. So to measure the head width, we have to train a deep learning model actually to recognize the head first and then segment the head. So that part might need some basic retraining if your organisms look very different from this, but everything else should be completely unsupervised. Yeah, as you can see, it works on a variety of body sizes and shapes. However, I always like to show where my stuff fails. So in this case, here, you can see that the length is going onto the antenna and on these appendages at the bottom. And you can tweak the parameters to avoid this, but there is always a trade-off between getting the common stuff working well and the stuff that is a bit weird also working well. You cannot have them at the same time, let's say. So we measure this, and the margin of error is around 5% compared to a person measuring on the image on average. Right, and just to go back to the biology very quickly, like if we run this pipeline on a couple of images that were never seen by the package, and we let it complete, basically, it will spit out something like a trait distribution. In this case, for the experiment that we had, these are different flow rates, and as you can see, the trait distribution, the body size, the length, is changing with the flow. So the strong flow will tend to bias toward smaller bodied organisms. So this is just a very brief example on how these techniques can speed up the work and get you to the data that you need and that the stakeholders need much more quickly. And this is like, yeah. But now into the more interesting, for me, part at least, since I come from a background in pollination biology, it's about biotic interactions. So biotic interactions are underpinning ecosystem services, and there is a lot more attention that has been given to how ecosystems work and what they provide that is important for society. However, organisms don't exist in isolation, but they interact with each other, so you need to study these interactions to be able to predict how the environment, the ecosystem is going to react to some changes. So very important functions of organisms' life cycle are underpinned by interactions. For instance, the survival, like mutualistic interactions seem to be more common than we previously thought in biology, but also pathogens, predators, as well as reproduction for plants, pollination, and dispersal. So basically, I'm trying to make the point that even though interactions have been understudied because they're difficult to study, they are as important, if not more important, than abiotic interactions for ecosystems. So yeah, here I say that they're difficult to study because they can rarely be replicated in the lab. They need to be in a natural or semi-natural environment. But in the field, in natural environments, the conditions cannot be controlled, so you end up with noisy and imperfect data. And pollination especially, I would say, is still measured by hand in a very time-consuming and error-prone way. So you basically sit out in the field with your notebook and note down every visit and how much it lasted and what was the behavior and how long it lasted, and etc., etc. So yeah, in 2 years ago, actually 3 years ago, I wrote a little review on the methods that are recording interactions in the field, and you wouldn't believe how this is outdated now because there has been so much progress in the field. But it's not a new thing that people start to put these cameras out in the field. It's just that the methods have been evolving so fast that nobody has been using the same methods twice, basically. Right. So this is a practical example of how you can use cameras to record interactions in the field. So this comes from my PhD. So I studied the evolution of the Asteraceae family in the Alps, and this is a species that is very common, widespread in the Alps. It's an Esodoroniko. It exists in 2 cytotypes. It's an octoploid cytotype that is very widespread and flowers a little early in the year, in a tetraploid cytotype. Cytotypes are just different amount of DNA in the nuclei, but otherwise they're the same species. This happens very frequently in flowering plants. And these tetraploid plants are much rarer and they flower a little later in the year. We have found a population in France where they co-cord, which is it's relatively rare for these cytotypes. They tend to also have some sort of ecological differentiation. So we applied pollinator monitoring to this population and see what was going on. So yeah, a little bit more about this plant is that they differ in a few other things, like the proportion basically of tubular flowers that are this one in the center, and these are the reproductive parts of the flower, and the ligulate flowers that are these long ones at the outside. And these are basically only serving like a vesiculiferous function to basically attract the pollinator, but they're actually sterile. And there is a different proportion between these flowers in the 2 cytotypes. And this is interesting because they're the same species and they're very, very close evolutionarily speaking. So it's interesting that they can diverge in these traits. And they also have quite a different amount of flower inflorescences per plant. Right. So the system that I used this was back in 2016, was made by a company called Tumbling Dice, and it's called Rana. So this is just a single board computer that is connected to a webcam and powered by a battery, and this will basically do something for you. So this is something, I will explain in a second. This is some images of the field deployment. So our field site was at 2,000 something meters, so we had to carry everything up every time. And it's a bit inconvenient to carry these big caravan batteries, I would say, up there. And yeah, this is our highly technological setup on how to affix the camera to the ground. And yeah, now I have to show you the video. Yeah, right. So, oh my goodness. Can we not do that? Okay. Right. So, this system is closed source. So I cannot tell you exactly how it works. But reverse engineering a bit what is going on. I believe that this is doing blob detection on these insects. So the thing that the system is doing is cropping out the frames where there is supposedly no visitor, but as you can see, there is still like the flower moving in the wind that is triggering the movement. And it's only compressing together in a video clip the frames where the pollinator is supposedly visiting. Another thing that I will point out is that we have to stake these flowers so that they stayed still, because otherwise basically it recorded continuously and it drained the battery in a very quick time. But the resolution is not too bad, and yeah, it was better than sitting in the sun for 14 hours a day. Pretty low bar. So yeah, so these are some results, but what this system gives you is videos. So I still had to look through probably 500 hours of video or something like that to score these images for what was the pollinator, how long was the visit, the identity of the pollinator and its behavior. So in this plot here, we can see that the tetraploids had a higher proportion of feeding visits, so potentially pollinating visits, than the octoploids. This is in spite that the octoploids should be more attractive because they have bigger flowers and they make more of them. So this is kind of counter to our prediction that we made. And yeah, and you can, since I also identified to some degree of taxonomic precision the insects that were pollinating these plants, you can then plot them in multidimensional space and say, okay, in the red we have like the morphospace of the tetraploid, and in blue we have the morphospace of the octoploid, and you then plot where the pollinators fall within these morphospaces. And even though these plants should be basically the same species and they flower basically at the same time in the same place, we can see that they are segregated in their pollinator communities. So the same species of pollinators don't tend to visit both plants, otherwise we would have points in between here. So even with these sort of like primitive systems for recording visits, you can still tell something quite important about the evolution of these plants and potentially the community of pollinators. Right, as I said, there are a few drawbacks, actually quite a few drawbacks, but there is no accuracy metric. It will only tell you there is something or there is no something. It does not draw bounding boxes, so you either get the frame with a pollinator or you get a frame without anything. And it does not do any classification, that's still a manual step, and it's closed source. It also, the battery, even though we used very big batteries, they only lasted around 5 to 7 days, so the power draw was really quite high, and it's very expensive. Like it's around 800 to 1,000 euros per unit, which is, considering the hardware, it's a lot of money. Right, so before moving on to the hardware improvements, I'll talk about like the second part of my previous project at VSL, and that's the hummingbird detector. So, brief introduction about hummingbirds. You probably know what I'm talking about. But hummingbirds are a fairly diverse bird group that live exclusively in the Americas, and they visit around 7,000 plant species in 68 families, many of which they entirely depend on. So they're a classic textbook example of coevolution, because plants that evolve the hummingbird pollination, they tend to evolve anti-insect pollination traits. So they tend to exclude the bees that would have ancestrally pollinated the genus of plants or the family of plants, and tend to only rely on hummingbirds. So there are good study systems for trying to study, for instance, the matching of traits between the flowers and the pollinator. Right, so the data that I've been working on has been collected in Costa Rica, Brazil, and Ecuador in a very large elevational span, and it has been collected with these off-the-shelf camera traps that don't exist anymore. The company is out of business and they don't make them anymore. But the thing that is important is that it records at 1 frame per second, so 1 picture every second, which is not a very high frame rate. And hummingbirds are really fast. So. Right, so some examples of the data that we're looking at. I put some circles so you can quickly see the hummingbird. These are some of the best examples of frames that we have. As you can see, the camera is pointed at a focal group of flowers, and you can have like the backdrop of like many trees or the sky, or looking a bit down at the ground. So it's a great array of different backgrounds, and they move because it's in the cloud forest and there is wind and there is changing light conditions. So this is a very difficult problem for computer vision to pick out the frame, like the object hummingbird from these frames. So yeah, these are some of the best images that we have. And now we move on to something that is a bit more typical, because as I said, hummingbirds are really fast, so they come and go quickly, and they sort of look like a blur, a colored blur in the images. And we also have these fantastic images where they assure me that there is a bird in here, but sometimes I struggle even now. And so yeah, these frames have been painstakingly annotated manually by a team of experts that are familiar with the hummingbirds, and they also have attached a taxonomic label to whatever degree they were able to identify it. So, yeah, I will quickly skim through this because we already heard a lot about this, but I guess the point that I want to hammer home is that machine learning models and neural networks are nothing more than big models with really a lot of parameters. There is no black box, there is no magic, there is no mystic, anything that goes on behind the scenes. They're just big models. So you basically provide training data, and you know the output that you desire, and you basically force the neural network to learn the weights that give you the desired output given the input. That's basically what happens. And deep learning is a subcategory of this that has taken over the accuracy metric scores, because it uses many layers of neurons, and at each layer they learn a different feature. And with the more famous networks, there is quite a bit of studies done. You can know more or less which features are learned by which layer. So that's useful, for instance, for optimizing your hyperparameters, if you care about that. Right, so coming back to the hummingbirds, we have the problem that less than 0.1% of frames contain an event of interest. So this is a very rare event and in a very complicated environment. So what we did is to train a binary classifier with a class there is a bird and a class there is no bird for hummingbirds visiting focal flowers. And we also did some short-term visual differencing that I will explain later to basically help the deep learning model better score the frames where the hummingbirds are present. So I say here that is a basis for a generalizable model that detects small visitors, but really our trained model only detects hummingbirds. If you try to apply it directly to another domain, it will not work properly. We give you the opportunity to retrain it, but as it is, it's overfit. It's exactly what Sarah was saying, like that ecologists should stop saying that it's 99% accuracy because it only works in the data set. This is exactly what we've done, but that's what we want to. So yeah, a little bit about the architecture. Well, it's implemented in PyTorch, which is quickly taken over from other frameworks like TensorFlow or Keras as like the go-to for experimenting and iterating in deep learning models. And we use the DenseNet 161, but we also implemented ResNet, EfficientNet, VDG, many other architectures, and they give very similar results. And yeah, we train a lot because we want to basically push the model to the maximum recall possible. So basically, the recall metric is how many positive frames that the model has seen in training it will remember in, it will correctly guess basically. It's much worse for us to guess incorrectly and miss an event of interest than to spit out many true positives, many false positives, pardon. Right, so yeah, another important thing that often happens with ecological data, we only have positive frames in annotations. So the people that went through the videos, they only picked out the frames that contain a hummingbird, but we never have the ground truth of this frame does not contain a hummingbird. So you have to be reasonably sure that the rest of the frames that are not marked as containing a hummingbird will be empty frames, will be your negatives. This was not the case, so I had to annotate a bunch more, but basically in general, what I suggest to do when there is like something, a situations like this, when you only have the positive frames, is to try to have a random sampling spread across all your sites, in all your datasets, in all your training, test, and validation datasets. Otherwise, your model will really struggle to try to understand what the background looks like if you don't provide it a diverse enough set of frames. Right, so we don't just use the entropy score, like the probability score, let's say, that the deep learning model gives us, because that is that's very variable, and it tends to go from very low to very high with nothing in between, so it's not very informative for us to just use that. So we basically created a very simple aggregated score to give us a better resolution about the presence of a hummingbird. So we have P, that in this case I weight at 0.8, but these weights can all be changed easily, and they can also be optimized per location and per video in the pipeline. And we add to that the prediction, the PDIF, which is the prediction probability between the current frame that the model is predicting and the frame immediately preceding it. The rationale here is that if the model is confident that there is no hummingbird in the frame I-1, and it's relatively confident there is a hummingbird in frame 1, we want to amplify that signal because probably something relatively big has changed in between these two frames, and we want to amplify that signal. We also use a simple triplet differencing, which is an average pixel-wise standard deviation difference. It's just an histogram matching basically on how these two frames differ visually. It will be clearer when I show the images. And we do that on a sliding window of 3 frames, but also that can be adjusted and can be optimized based on your dataset. So a bit more visual explanation about this. So here we have the triplet differencing. So this is our current frame that we're looking at, and frame preceding it and frame immediately after it. And if you just match, like make the visual difference in between these two, you will find that the hummingbird, which is hopefully the thing that is changing the most between these adjacent frames, is giving a signal compared to the background. So we can assign a numerical score to this on how much this is changing compared to triplets of frames that don't contain a hummingbird, and we can amplify the signal with that. Another thing that we invested is, well, this is just a GradCam or a saliency map, and we wanted to see what the model was looking at basically to making the decision whether this contains a hummingbird and not. Ground truth class 0 is there is no hummingbird, and here we see that around the hummingbird that is here, it's looking at more the edges of the images and the surrounding of where the focal flower is. But where for ground class 1, the model is learning actually the pixels that contain the hummingbird. So it's not, we were concerned that it was learning some artifact about the data and learning more about the time of day or some illumination conditions rather than actually semantically understanding where the hummingbird is in the image. So yeah, so these are some examples of how it looks like. Here we have the hummingbird in our current frame, and you have a visual differencing. You can see that it was present in 2 frames here. So you have like basically a ghost here and like the more clear signal here. And yeah, we have a 99% probability for ground truth 1, so there's a hummingbird, and the same goes here, basically. Okay, so this is another visualization, and so basically this is like the score of all of the scores throughout all the frames of one video. One video is around 25,000 frames, and the red triangles looking down are positive events that were manually annotated, and black triangles are negative events that I manually annotated to make sure that this really didn't contain any hummingbirds. You can see in orange the deep learning model only score, and you can see that it's not jumping out particularly from the background on its own, and neither do the differencing and the triplet differencing on, like, they don't give very sharp peaks, which is exactly what you wanted to be able to pinpoint. Like in this frame, there is a big difference in probability score between the adjacent frames, and but the aggregated score, like the weighting that I showed you before, really helps you sharpen these peaks and be able to tell apart the probability densities of these from the other. So this is kind of a good scenario in which it worked well for this location in this video, but and I show you here some accuracy metrics. So as I said, I wanted high recall for the classes, for the hummingbird class, so we have 1 recall. However, as Sarah says, this is overfit. We know it's overfit. We trained it like a ton to make sure to have 100% recall, because it's imperative to have all the frames that contain a hummingbird in this. What we basically do with the aggregated score is, so the deep learning model only guesses correctly all the frames in which there is a hummingbird, but it spits out quite a bit of false positives as well. The only thing that we really do with the aggregated score is cutting out a bit the number of false positives, so increasing a bit that. So yeah, I also like to show somewhere where my approach kind of fails a bit more. So this is another video in which the aggregated score really does not help you all that much in distinguishing what is a true event and what is a false positive. So there are drawbacks to this approach as well, but that's the best that we found so far to extract this information from extremely complicated and changing background conditions. So yeah, there are some more scores for that. So the other thing that we provide you the possibility of doing basically is selecting the number of frames that you want the model to return, and if you train on your own data, it will give you these curves, so you will so you know that if you say, okay, I want the 100 top scoring frames, and you will have these dispersion for the probability only score and the confidence score only. This seems like a trivial thing, but when you have field assistants or annotators, you really need to budget the time that they spend on anything, and if you know the amount of time they spend on each frame, then you only give them an amount of frame that you have budget for, basically. Right. So this is not released yet. We're finishing up packaging it, and it is not a general model for visitor detection. However, we hope to be developing this more in the next year, and we use it. We provide different checkpoints at different different numbers of training epochs. So the model is you basically can choose your level of overfitness of the model. You can choose something that is completely overfit on hummingbirds, or something that is just starting to understand how to detect visitors in this kind of environments, and then train from that checkpoint on your specific data set. We package it again as a Docker container, and you can easily retrain it in a modular way like we showed you for the macros of Ventos. The other neat thing about the collaboration with the Swiss Data Science Center is that they have a nice tool that basically helps you to versioning your data, the experiments and workflows in a very easy way. Basically, it does it for free, but you don't have to use it. It's just included in the data. Right. Another very interesting development that I would like to implement in the hummingbird detector as soon as possible is using variational autoencoders to detect anomalies in video, because when the events are so rare, we're basically looking for an anomaly. So autoencoders basically try to have a latent space in which they construct a representation of the image, and you can also reconstruct the image up from this latent space. It's what is used in video compression, for instance, to save some space. You can also use them, train them on negative frames that don't contain hummingbirds, and try to predict the following negative frames given one video, one location, whatever have you. And if the reconstructed frame is very different from the actual frame, you can treat that as an anomaly and try to implement that as an additional score, basically, to help you with determining to bring down the false positive ratio, basically. Yeah, but this just finished, so we would like to experiment some more before releasing it. Right, so I'm not the only one doing these things, so I will give you some other examples that have been out in the literature. So, oh, actually no, first I talk about this other extremely basic project that we've done. So, we're interested in monitoring insects, not necessarily only hummingbirds. We developed this on hummingbirds because we had a lot of annotated data and a lot of expertise in-house to do that. But to monitor insects, it's sort of a different story. And we tried to test off-the-shelf camera trap models. We tested 16 in total with a wide spectrum of price. And I selected them to have very different specs. And then I tried them, and then I opened them, and they all have the same sensor. I wrote to the manufacturers, and some of them, they openly admit that they're the same sensor and the spec sheets are wrong. So I don't know what to make of that. Like, they literally declare that they can record clips of 60 frames every 10 seconds with a pause of that, and they don't do that. They all do clips of 5 frames every minute or so. So this is like, I don't know how is it possible that an industry has taken this direction, but apparently this is where we're at. And the other thing that is extremely important is that they don't have adjustable focus. So they're all quite far away, and when you have something small that you want to be close to so that it occupies a reasonable amount of the image, this is kind of a problem. Right. There are claims in the literature that camera traps are adequate for monitoring insects. I put this paper, but it's not the only one. But I yet to see any that include comparison with human observations. So if you see a paper with camera traps and ectothermic organisms that don't emit heat, so they don't trigger the camera trap, maybe you should be skeptical. Yeah, we did some amount of testing in 2021, and they miss always more than 50% of the events compared to a human. And already the human misses a lot of events because it can't look at the plant at all times. Yeah. So what we did is to go to the literature, and we found that this nice paper that used a Raspberry Pi based system. I took that protocol and tried to improve it for efficiency, so the software is less flaky and it's a bit more easy to build. Everything in this system can be built from things that you can buy off Amazon or whatever, and you don't need to solder anything. We managed to push it to record 14 hours a day for more than 10 days on a single charge, which I think is pretty reasonable for an ecological monitoring program. And we currently don't have any processing happening on the device. We just record the frames. And this is often very important because we really don't know what pollinates a lot of things. So if you don't even know the size, the sort of functional yield of your organism, it's really hard to pinpoint a time of day or other characteristics that might help you to yeah, and it costs less than 200 euros, but if you're really cheap, you can make it for around 70. So this is how the system looks like. I realized that I took very little pictures of my actual systems in the field. So this is actually the picture that comes from the paper. The only thing that we do differently is that we use a different version of the Raspberry Pi camera, but there is no other difference basically. So if you're not familiar with Raspberry Pi, the operative system lives on the SD card that it's inserted in the Raspberry Pi. And I provide a precompiled image that you can just flash to it. You connect via SSH, you just have your phone, you have an hotspot, you connect to the Raspberry Pi, and with an app that is free, you can just connect to it and interact, basically, without cables, without having to open the casing. When you go to the field and it's been out recording for a while, you just access it, turn it off, open, retrieve the battery and the SD card, put in a new one, and then download the data when you're back in the office, and then you can do all your post-processing. Right. Currently, we have 60 of these cameras being deployed in Ecuador, and they're quite hard field conditions because the temperature goes up and down quite a lot, and it rains and all these kind of fun things. But we also tested it in the Alps last year. So we had basically the double aim of collecting sample interaction across communities and collecting the data for actually training the detection model on insects, starting from the one that we did on hummingbirds. We monitor two species of plants. One that is a generalist, so it's pollinated by a wide array of things, and one that is more specialist, and it's only pollinated by bumblebees across an elevation range. So the idea here is that with climbing worming, species can migrate up the elevation gradient, and insects are being shown to be faster at migrating up, and this creates a potential mismatch between the flowering time of the plants and the emergence of the insects, which is bad for fitness of both of plants and insects. Right, so this is some pictures of our deployment. We tested our prototypes in relatively harsh field conditions. It's around 2,400 meters. There is high UV, so it discolors everything that it touches, and it hardens rubber, plastic, and a lot of other things. And the temperatures ranged from -5 to 35 degrees, and there is very strong winds all the time, basically. So it needs to be quite rugged, the system. These are some examples of the video that you can collect. These are zoomed in on 1/4 of the frame, so the video is much larger. And you can see that, again, these are small plants, there's around 10 to 15 centimeters tall maximum, and the quality could be better, but I'm limited by the amount of processing of the video that the Raspberry Pi is able to handle at those conditions. Okay, so we end up with a ton of data, and I am only one person, so I can't annotate all of it. So what we did is make a citizen science project on this portal, Zooniverse, which has been mentioned before. And this is also the chance for me to show you some pictures and how difficult it can be. So I just logged in, and so we can look at some pictures. And the idea here, this is an annotation tool that everybody can sign up to and try to annotate the pictures. Here you select the speed of playback, and I provide five frames for each. So here you see, it's kind of funny because there is probably a person or a cow on the frame, but the idea would be that if there is an insect, you say yes. In this case, I don't see any, so I would say no, and I say done, and it provides the next set of images. So here, for instance, you can see that the camera moves, and this is something that we want to improve, because you don't need the movement of the camera to deal with, as well as the background. So, I also don't see an insect. I have to say that probably we won't see an insect because it's quite uncommon in these images in alpine environments. But yeah, this is basically to give you a flavor of what it's like to annotate this kind of data. And the other thing that I wanted to show you is that if you give people the chance, a lot of them will sign up. Like, I want to say that around 50 to 100 volunteers are working on this, and if you provide feedback to their problems, they really are eager to engage even with these relatively complicated problems. The other thing that I want to point out is that Zooniverse has this policy of having more than one person look at the same frame, so you can set what is called a retirement limit before this set of frames is taken out by the pool. And we set it really, really high to 30, so 30 people will look at the same set of frames, because the annotations are really bad quality. So like the bounding box that I asked to draw around an insect can vary. I think the last one that I ran was around 150%. So it's really like you have to treat them, the data that you get out of citizen science project, as raw data that you need to post-process to be able to then feed it to a machine learning algorithm. And you still need to treat them as weak labels. So, okay. It was a bit about the, yeah. And these are some random numbers if you want to run cameras in the field. This is sort of like the file sizes that you expect. This is something that I never found written anywhere, so I just put it here in the hope somebody maybe in the future will use it and it will find it useful. And the other consideration is that since I work on this system on both on the hardware side, improving it a bit, and on the software side, making the scripts easier, the protocols, in order for field crews to be able to use it, needs to be very well written and very detailed. And even if you do that, these are simply Word files, because anything else basically they didn't want. And even if you do that, sometimes they decide that their way is better, and they want to open the box every time that they visit to see the video, and I find that not very useful compared to having a phone that shows you the video. But what I'm trying to get across is, even if you develop something that you think it's really good for the task, and other people do something else, don't give up, because otherwise this data would have never been collected. These sites have never been sampled before, and there are extremely rare species of hummingbirds that I've never seen before. So interacting with a flower, I should say, not new to science. So yeah, it's my encouragement to all the tool developers, don't give up, even though it's always frustrating a little bit. Right, so other examples of applications. This is the diopsis system that is being developed by a collaboration with Naturalis in the Netherlands. And what we do is they detect insects that land on an attractive surface. In this case it's yellow, but it could be a different color, for instance. And it does the segmentation and some amount of classification on its own. Another two traps that use the same principle is this, that has been just published, and it has this plate, and it detects the insect flying in from this on this plate. And I guess it's still called the Arus moth trap. I don't know how else to call it, but it's basically a trap that illuminates a sheet where the moths at night will land, and it will do the segmentation and some amount of classification. It's been developed at Arus. And as well, this may be currently, as far as I know, one of the more advanced systems that you can have. And this is real-time monitoring of and tracking of the insects flying onto a small patch of flowers. The limitation of this is that it does the segmentation and the classification at the same step with a YOLO framework, and so it's therefore limited by. So if you have many classes, you will also struggle to train this. Right. So this is currently my new project that I just started, so I don't have much to show, but we are basically trying to transfer what we learned on hardware and on detecting hummingbirds and then starting traits from images to a system that can be put in the field and will do some amount of these things on its own. So what we are trying to do, especially on the hardware side, is trying to use a device that has an integrated GPU on it and can run simple machine learning and can do the segmentation in the field. So my objective would be to have at least the detection to cut out a good model for detection, to cut out most of the frames, and then be less constrained by the storage capacity. Right, so challenges, of course. Training and deploying models requires specialized know-how, so there is a high barrier to entry for ecologists to be able to even use these tools a lot of the time. And the point that we made also yesterday during the discussion was that the open data and model standards for ecological applications are non-existent, so it's really hard to compare apples to apples because everybody is doing something slightly differently. And we also saw that model developed for one ecosystem transfer poorly to other ecosystems, even though they superficially look similar. Of course, natural conditions are hard for CV models, and the impact of different hardware used to record and different models is also not clear on the quality of your data. Right, this is basically the same stuff, and in the spirit, yeah, I'm just gonna skip this. In the spirit of building community, what I did is to put some useful resources so that in the talk, so you can just go and find them. This is Wild Labs. Wild Labs is an ONG based in Britain somewhere, I don't remember, that their job is to connect researchers working in conservation, biodiversity research, ecology, stakeholders, all kinds of things. So you can sign up for free, and there are several different groups, interest groups that you can join, and you can get feedback. We really should be a little bit more active, but we're all on there, basically, a little bit. The other thing that I will mention is Lila.science, which is a massive undertaking by Dan Morris, and it's basically a giant repository of machine learning ready annotated datasets for ecology. So if you are a developer and you want some ecological data to play with, this is the place to go. There is a ton of stuff on there. And then it's been mentioned before, I put the link here as well, the Slack channel for AI for conservation that Sarah curates. Slack is a little bit stupid. You have to send an email to a certain address to get the invite, but that's automatic. You get the invite back, you just click on the link and you're there. Right, and me and Moritz have already organized another mini thing like some years ago, and as I mentioned to some of you, it's increasingly easy to get sessions about automating monitoring and computer vision methods and deep learning methods in conferences. So I really do encourage to go to your domain conference and say, okay, I want to talk about these things, and you get a lot of people coming to your conferences. And there are increasingly some schools. So finally, there is a cost action starting on exactly monitoring insects, which, if you're not familiar with, a cost action is basically a networking grant from the European Union that allows people to meet each other, organize meetings, organize summer schools, and these kind of things. And it's just getting started now. It's very bottom-up, grassroots, so you can join and contribute, and there is no structure as of yet. We still have to start. But I really do encourage you to go, and there is a lot of the important players in the space now there. Right, with this, I'm done. I thank you for your attention, and yeah, please ask any questions.